work. These include
Very happy to be here today. It's a beautiful morning in Minnesota. I hope that wherever you happen to be in the world, it's also a beautiful morning for you. And I'm really excited to talk about, as Dr. Knights mentioned, some methodological work that we've been doing to try to improve the use of metagenomic data for surveillance of antimicrobial resistance. Before I launch into the talk, I really have to acknowledge my close collaborators who have done a lot of the work that I'll be presenting today. Dr. Christina Boucher is a computer science associate professor at the University of Florida. She works on bioinformatic algorithms, and she and I have been collaborating for several years on this topic. Dr. Ilyas Lazovsky is currently a dual PhD residency fellow in the College of Veterinary Medicine here at Minnesota. And he also made major contributions to the third part of the talk that I'll talk about today. And so these two individuals are just crucial. I wouldn't be here talking to you today without their work on this important topic. So thank you to them. So the first thing I want to do is set the stage. I think this is probably a fairly diverse audience. It usually is when we're talking about bioinformatics. We have people from multiple disciplines. And so I think setting the stage helps to get everyone sort of thinking in the same context, which then will lead me into the science that we've been actually doing in this area. Antimicrobial resistance is considered one of the top global public health threats facing our world today. This is just a snapshot from the CDC's page about AMR, or antimicrobial resistance. And as you can see, tens of thousands of people in the US die from antibiotic-resistant infections every year, and that number seems to be increasing over time. And so what that means basically as a person is that if you get a bacterial infection, there's a higher likelihood that potentially that pathogen may not respond to antibiotics the way that a human doctor would hope. And that's the same for animals as well. So I'm a veterinarian, and we worry about that in the animals that we treat as well. So we've known for a long time that this is a potential problem. And in the US government has released the second phase actually of what they call the National Action Plan for Combating Antibiotic-Resistant Bacteria. This is a very long document. It details a lot of things, a lot of aspirations that we should strive to accomplish in order to combat antibiotic resistance. I've listed the 5 main goals here on the right-hand side from this National Action Plan, and I've highlighted the second goal, which is to strengthen what they call One Health surveillance of AMR. One Health is a term that we're very accustomed to in veterinary medicine, and what it refers to is the fact that the health of humans is actually interconnected with the health of animals and the health of the environment. So it's sort of this integrated system of animals, humans, environment, and there are lots of diseases and other things that fall within that intersection, but AMR is sort of a classic poster child for the One Health concept. And that's because these resistant bacteria and these resistance genes, of course, don't only impact humans, but they also impact and reside in animals, and then also within the environment. And so if you really want to talk about controlling AMR, you have to talk about controlling it across all of these different sectors. So surveillance is this concept that we want to establish a systematic sort of snapshot of AMR over time and across the US so that we can understand trends in AMR both over time and across space. And in the US, we've had a long history actually of antimicrobial resistance surveillance. So I put here some pictures. NARMS is a unique collaboration between FDA, CDC, and USDA, where they take samples on an ongoing basis for a couple decades now, and they look at resistance trends. The National Health Care Safety Network is a human hospital-specific program. USDA FSIS focuses on pathogens and antimicrobial resistance in livestock and poultry products. And then there's also, of course, numerous companies and other smaller public programs that focus on surveillance for AMR. So in these surveillance systems, the historical context is that in order to surveil for AMR, these programs have focused on phenotypic AMR in specific bacteria. And so that's what we call using a culture-based approach. And so I represent that here just by a culture plate. And, sometimes these surveillance programs will target pathogens like human pathogens such as salmonella, but other times they'll target what we call indicator bacterial species. These are species such as generic E. coli that may not be pathogens, but they're very easy to culture. They're ubiquitous, and therefore it's easier to conduct AMR surveillance on them. And so what they do is they grow these indicator bacteria in the presence of different antibiotics. And if the bacteria are able to grow, that's an indication that they may be resistant to that antibiotic. And that's how the surveillance is conducted on the ground. In reality, thanks to non-culture based approaches and specifically high throughput next generation sequencing, we know that when we're looking at these samples, they don't just contain these indicator bacterial species or pathogens, but they contain thousands of bacterial species. And that's a consideration when you're trying to talk about AMR, because not only do the pathogens and indicator bacterial species carry AMR genes, but these other bacterial species can also carry AMR genes. So the underlying assumption is that this wider microbiome is important for AMR, and I'm going to make the case now that I think these other bacteria do matter. So even though, of course, pathogens are the major concern when we're talking about antimicrobial resistance from a clinical perspective, I think from a public health and surveillance perspective, these other bacteria are also important, and their AMR profiles. And I think there's a few reasons for that. First is that when we look at indicator bacterial species, we're sort of assuming, it's sort of an unspoken assumption, that AMR patterns in those bacteria are sort of a representation or a proxy for AMR trends in the whole microbiome or the whole microbial community. And that's not necessarily the case. And so, if we're trying to understand trends over time, we need to be looking at a representative portion of the bacterial population. The other thing is that the commensal bacteria can be what we think of as a reservoir of AMR. So they can harbor these resistance genes, and bacteria can undertake horizontal gene transfer, whereby they can move entire sets of genes and genetic material, even across distantly related bacteria. And so that mechanism is very important for bacterial evolution, and it's very important for AMR. And so if you have AMR genes in these commensal bacteria, they could find their way into pathogens, for instance, within your gut microbiome. And then the third point here, I think, is a little more nuanced, but when we're talking about surveillance and trying to intervene and control the flow of antimicrobial resistance from this One Health perspective, that's a really tricky thing to do, and we really need additional data points. And my hypothesis is that these other bacteria and their AMR genes can provide actually additional data points to allow us to conduct surveillance in an improved fashion. And the reason that's important is because tracking antimicrobial resistance is really hard. So I'm an epidemiologist, and I think about this sort of from a population and systems perspective. And so the way I think about it is, thinking about all these different One Health type spheres within our society that we use antibiotic drugs, and we place selective pressures like anthropogenic selective pressures on microbial populations. And so, obviously in human habitats, we do that like hospitals, but other areas like aquaculture production, in our companion animals, we often give them antibiotics. So there's all these different areas, food animal habitat, so livestock production, all these different areas where we're sort of placing pressure on microbes. And the real goal is to understand how resistance is developing in each of these spaces and how it's then transmitting between these different sectors. And that's where it gets tricky, because that's where you have to think about all of the very complex pathways, and a lot of them environmental, like water, wind, soil, but also the food chain, and then of course direct contact, to understand how antimicrobial resistant bacteria and also the genes that they're carrying are moving around. And this has been, a challenge for a very long time. It's still a big challenge, but I think that the use of a microbiome-wide resistance profile could help gain some resolution on how resistance is disseminating and transmitting within these complex pathways. So now that I've hopefully convinced you that it's likely that this microbiome-wide resistance profile is important, I want to talk about how we're currently typically generating and analyzing these microbiome-wide types of data sets. And that will set the stage to understand where there are current limitations and how we're trying to tackle some of those limitations. So I'm going to start with what we call the standard workflow, and I term it a shotgun metagenomic workflow, and it goes something like this. So we start with a sample, and that can be any sample, any type of sample. We extract the DNA. This is a very standard workflow that many groups and labs and also regulatory agencies are starting to do across the world. So we take the sample, we extract all the DNA, fragment it into small pieces, perform library preparation to then sequence it on typically a short read, high throughput sequencing platform such as the NovaSeq 6000. That sequencing platform then generates sequencing reads, which are typically short, 200 base pairs or less strands of the sequence of DNA that's in that sample. So then, of course, the goal is to take all of the sequence data that we generate and to find the resistance genes, because we're interested in surveillance of resistance. And in our lab, we have a reference database that we use to do some of that work. So the database is called MegaRes. And in that database, there are the sequences of over 4,000 known antimicrobial resistance genes. And we have then annotated each of those genes within a hierarchical acyclic graph structure, which allows us to perform some different types of gene finding algorithms, such as hidden Markov models and other machine learning algorithms, to hopefully improve the accuracy of detecting which resistance genes are in a given microbiome and how many of them are there. And so we then perform this process, and then we have basically a list of all of the resistance genes and their annotation within a given metagenomic data set. And then we need to take that and do what I'm very broadly going to call resistome analysis. And so resistome analysis can look like a lot of things, but at a very basic level, it's a description of the resistance profile in a given set of samples. So I'm going to give you just some very quick sort of ideas of what that looks like, again, to set the stage for methodological improvements. So the first thing we can do, of course, is we can describe, right? We can look at what genes are there. So this is a study we're currently conducting where we're taking fecal samples from 108 pigs as they age. And so these are 6 time points. Each of these little columns is one pig sampled 6 times over their lifespan. And what you can see is that, we see this variability in the resistome profile. It tends to stabilize at a certain point, and it looks fairly similar between all of the pigs in this population, right? So this is a way to understand at a high level trends in changes in the resistome in individual animals over time. Of course, if we can describe, we then quickly want to compare, right? So if we have groups of things and we can describe what resistome they have, then pretty soon we say, well, can we compare them? And of course we can. So this is another example of a way to compare resistome. So in this study, we sampled animals when they arrived at a livestock production facility, and then again when they exited the facility. So those are those black columns in the middle of this figure. And we basically described which of these resistance mechanisms were prevalent at what percent prevalence at arrival and exit. And so in this population, you can see that at arrival, they come in with a fairly diverse resistome. And then when they exit the facility, the resistome is less diverse. And some of these resistance mechanisms seem to have disappeared from the microbial population. So this is a way, again, to compare shifts in the resistome. We can also do that using ordination, which is also very common. I'm sure many of you have seen this. So this is, again, a group of animals where we gave them a certain treatment, and we took samples before and after the treatment. And each dot here represents one of the resistome, one of the samples from these animals and its resistome composition. And you can see that the samples taken before treatment cluster together and separately from the samples taken after treatment. And so this, again, is a way to compare the overall resistome composition between groups of samples. So, you know, this is useful, and people do this a lot, these comparisons and these descriptions, but there are some really, when we think about antimicrobial resistance surveillance, which is sort of a different use case, there are some fundamental limitations to this current standard workflow, going all the way from the lab through to the statistical analysis. And I'm going to talk today about three of these limitations. Well, actually four, but I've lumped two of them together. So three buckets of these limitations. And those are sensitivity, scalability, and accuracy and context. So I want to start with sensitivity. So sensitivity is the ability to detect a resistance gene when it truly exists within a given microbial community in a given sample. And the real fundamental limitation that we deal with is that for most of our samples, the resistance genes comprise less than 1% of all of the DNA. And so what that means is that we are sequencing through a whole lot of DNA that we're not as interested in before we're sort of quote unquote finding the resistance genes. And that can lower our sensitivity potentially. And the first time we really were confronted with this in a very obvious and stark way was when we had a set of samples and we had both culture-based results, PCR-based results for some specific resistance genes, and then metagenomic data as well. And so we had all three types of resistance outcomes for the same set of samples, and we published separately. This took place over time, these three different ways of looking at these samples, and we published these papers. And then when we thought about the results, we realized that they were not very comparable. And so these are the three papers that came out. And what we saw at a very high level is that when we were identifying some resistance genes on PCR, we were not identifying those in the resistome, even when we performed some additional bioinformatics to really try to pinpoint some of these genes if they were there at a very low level, but they were not. And so that was a lack of sensitivity, in our opinion, from the resistome data. We also were looking in this set of samples at some associations between antibiotic exposures and the resistance profile. And when we used the culture and PCR results, we did identify an association between the use and the resistance. But when we used the resistome analysis, we did not identify that association. And so again, we wondered if that was because there was a lack of sensitivity to detect some of those resistance genes within the metagenomic data. So we wanted to try to solve or increase the sensitivity, solve this problem to some degree. And our solution was to do what we term custom targeted enrichment or probe enrichment, also termed bait enrichment. There's many names for this, but basically the overall goal is to increase the percent of on-target sequencing by enriching for the targets of interest before you sequence. And so the concept at a very high level is that you design what we call baits or probes, and those then bind to the fragments of DNA that they match to within the soup of DNA in your extracted DNA. And then you amplify up the bound DNA, you wash away the unbound DNA, and then you have what you hope is a relative increase in the proportion of your targets of interest within the DNA. And so the first time we did this, we used a design of baits where we targeted those 4,000 resistance genes within the database that I mentioned. And there were 50,000 unique baits needed to do that. And of course, the question was, when we did this enrichment, would it indeed increase our ability to detect resistance genes in the sample? And the way that we tested that is we took a set of samples and we did just the standard workflow without enrichment. And then we took the same samples and we used this enrichment process. And at a very basic level, what we found was a 3 to 4 order of magnitude increase in our what we call on-target sequencing. So this is a simple dot plot where we have the set of samples non-enriched on the left-hand side, and then the same samples with the enrichment on the right-hand side. And the y-axis is a log scale of the percent of on-target sequencing. So you can see for those red samples, which were wastewater treatment plant samples, they have very, very, very low abundance of resistance genes to begin with. So down at the 0.00 0.01% basically. And then when we enrich those, we get up to maybe 1% or less of the DNA. For some of the samples that had a higher starting proportion of resistance DNA, maybe around 1%, we did get over 50% of the enriched data being what we call on target. And so this was promising because, of course, this is what you hope, is that you're increasing your on target sequencing. But the question was, did we find anything new, or did we increase our sensitivity to detect resistance genes with this on target sequencing? And indeed we did. So this is actually a Venn diagram, but it looks like a hard-boiled or like a soft-boiled egg, because when we looked at the non-enriched resistome in this set of samples, we found 203 unique resistance gene groups. We re-identified those 203 resistance gene groups in the enriched data, and we found an additional 246 resistance genes that had not been detected without that enrichment step. And so we more than doubled the diversity of the resistance genes that we were able to identify in this set of samples. And importantly, for those of you who are involved in resistance work, some of those additional genes that we were able to detect were genes of high importance, such as ESBLs and carbapenemases that I've listed here on the right. So, you know, those genes were there. They were there in the DNA. It's just without the enrichment, we weren't able to detect them. And so we think that this enrichment method is one way to increase the sensitivity of this resistome analysis. And so that we feel is a methodological advancement in this area. So now that we've talked about that, I want to move on to scalability and accuracy. So one of the challenges with resistome, well, with metagenomic data broadly, is that these are very large data sets, and they're very complex data sets. And that can pose some challenges for existing bioinformatic algorithms. And so really the problem is the size and nature of metagenomic data, which often surpass sort of the practical limits of what we can do from a bioinformatic standpoint. And, you know, why does this matter? Well, you know, for surveillance in particular, we're talking not about, you know, tens or hundreds or even like low thousands of samples, but we're talking about potentially hundreds of thousands of samples on an ongoing basis. And so most of the studies that I showed you, and many of the studies in the literature, sort of max out at around 1,000 or a few hundred samples in terms of metagenomics, but we need to be able to handle many more than that. There are also thousands of known AMR genes, and in every sample that we generate from a surveillance perspective, we need to search for all of the known resistance genes, right? So it's an iterative process of always trying to find the same sets of genes in every sample. Metagenomic samples are tricky also because there's lots of homology between these resistance genes, but also at the same time, lots of variation at the nucleotide level. And so that can pose a problem for some of the bioinformatic approaches that we typically use, and I'll talk about those in a little bit. And then ideally, you know, in addition to just finding things that we already know exist, we'd also like to be able to detect novel variants, because one of the goals of surveillance is to see if there's, you know, emerging resistance in certain areas. So our solution to this was what we term a succinct colored de Bruijn graph, and then an extension of that, which is a read colored de Bruijn graph. And so I'm going to talk now very briefly about de Bruijn graphs and their importance for metagenomic assembly. So the de Bruijn graph was first described in the context of assembly in the late 80s, early 90s, and really sort of came into its own in the early 2000s. And that's because at around that time, so previously assembly had been done primarily using OLC, so overlap layout consensus approaches. And those approaches really require all pairwise comparisons to be performed on a given sequence data set. So when you think about not first generation, but next generation sequencing data, you have millions and often billions of reads, and performing pairwise alignment across all of the pairs of reads within that data set becomes just limiting from a memory perspective. And so the de Bruijn graph was an attempt to overcome this barrier of the OLC approach and have a more efficient way of reconstructing the actual DNA in the sample from these fragmented sequence reads. And so I'm sure many of you on on this call today have used some of these assemblers. There are many more of them. They're continually improved upon and, you know, sort of modified for different use cases. I'm going to now talk briefly about the very high level approach to constructing the De Bruijn graph, which will lead into our methodological improvements. So the basic idea is that you choose an integer value of k, and then for each unique k-mer that exists in any sequence within your metagenomic data set or any sequence data set, you create an edge for that unique k-mer. And then the preceding vertex is labeled with the prefix of that k-mer, and then the suffix vertex is labeled with the suffix. You then look at all of the vertices across this graph, and you glue together vertices that have the same label. And so what that can look like is this. So say we have a short input sequence, and we have we're choosing an integer value of k of 5 in this instance, so 5-mer. And you can see here we label the edges as ATTCG and ATTCA. The prefix vertex is ATTC in both cases, but the suffix vertex differs by 1 nucleotide. And so in the construction of the De Bruijn graph, you would glue together these vertices, and then you would have this branching node within the graph. So this is a very simple example. This is an example of of a larger De Bruijn graph looking at the entire sequence of T, which is represented at the top here. And you can quickly see that these graphs can become very complex, and then traversing the graphs to reconstruct the original sequence also increases in the amount of time needed to perform that task. So a color De Bruijn graph, to compare it to this standard De Bruijn graph, is where we have at least 2 input sequences. In this case, we have T and S, and T and S differ by just one nucleotide. And we have what we call a union De Bruijn graph here at the bottom, where we have merged the individual graphs for T and S into one graph. The paths in the graph, the vertices and the edges in the graph that are common to both T and S are colored in purple, and the vertices that are specific to sequence T are colored in blue, and the vertices that are unique to sequence S are colored in red. And so by using the coloring of the edges based on the input sequence, you can, in one graph, you can tease apart variants that are occurring within your input sequences. And so from a metagenomic perspective, this is important because we're putting a diverse community of sequences into the De Bruijn graph, and we want to understand where there may be variation, but also homology, right? So that's where you can tease apart areas of variance, or the bubbles we call them, and then common paths where all the colors merge together. So this was described in 2012 by Iqbal et al. And the problem with this approach in this construction is that it's very memory intensive and also time intensive. And so when we're thinking about applying this to 4,000 resistance genes, each of which need their own color, and then hundreds or thousands of samples, we quickly run into just surpassing the limits of RAM that you would have in even the largest computer available. And that's the case because partially because when you think about a metagenomic data set, you probably have on the order of tens of billions or hundreds of billions of k-mers for an individual sample. And then of course, thousands of colors to represent each sample and each gene. So what the improvement that we decided to try to do on this was to take the current method of storing this colored De Bruijn graph and making it more efficient. And so this is a paper from, this is a central figure from a paper that my collaborators and I published in 2017, where we have sort of the graphical representation of this colored De Bruijn graph on the left, and in the middle we have the sort of standard way for storing this constructed graph, which is a colexographical ordering of the vertices. But again, in this construction and in this storage approach, you need 8 times the number of k-mers in terms of bytes. And so if you think you have like tens or hundreds of billions of k-mers within a metagenomic sample, you'll need 8 times that in terms of bytes of memory. And so you can see that quickly becomes infeasible. And so what my collaborators, and this again is Dr. Christina Boucher and her group really pushing forward on these algorithmic advancements. But what they did is they took this lexicographic ordering and they turned it into a Burroughs-Wheeler transform on the edges themselves. And so that's represented over on the right. And so now instead of 8 times K, the number of k-mers, you actually just need 4 bytes per k-mer. And so it's a massive reduction in the amount of memory needed to store the graph. And then also you can, because of additional bit vector arrays that are created in this process, you can also query the graph for specific k-mers in a very time efficient manner. So this is just one table of a result comparing VARI, which is the method that which is what we call this sort of efficient method of the color de Bruijn graph, and Cortex, which is the Iqbal 2012 incarnation of the color de Bruijn graph. And you can see that for the 5 data sets that we did this test on, the plants, the 3 E. coli data sets, and then a metagenomic beef safety data set, we actually weren't able to hold those data sets in memory using Cortex. And so we weren't able to generate like a comparison of actual memory and runtime. But you can see using the plant data set that, we go from needing 101 gigabytes of memory down to 4. And so that is a real space saving efficiency of this succinct Burroughs-Wheeler transform way of representing the color de Bruijn graph. We still have one sort of computational issue to try to resolve, which is accuracy. And one of the reasons we have a problem with accuracy is because when we have a metagenomic sample, as I said, we have all of these short sequences coming into the de Bruijn graph construction, some of which have a lot of homology. And so those regions of homology pose a real problem for a de Bruijn graph. And we also, in the de Bruijn graph construction process, we fragment those even smaller into k-mers. And so you actually lose the coherence of each read within your data set when you do that. And so then when you try to reconstruct full-length genes, you often have errors in that assembly of each gene. And so the way that we wanted to solve this was actually to extend the concept of that efficient colored de Bruijn graph by actually coloring each input read. So now instead of coloring each sample and each gene, we're actually coloring each read within each sample. And the reason we can do that is because of the efficiency gains that were made in VARI. And so we call this a read colored de Bruijn graph. And when we perform this read coloring algorithm, which maintains the read coherence of each input read, we are able to increase the accuracy of finding and accurately reconstructing those resistance genes within a metagenomic sample. So this is just one result of that, where we have 4 different simulated data sets across the top with varying depth of sequencing and coverage. And you can see that VARI, which is the read colored de Bruijn graph, consistently achieves high sensitivity and precision in finding resistance genes within these 4 data sets. In addition to accuracy, this read colored method also allows for efficient reconstruction of variants within the resistome. So this is just one example where we used VARI to find variants within the resistome for 2 samples, sample A and B. And this is the TET-L gene within this metagenomic data set. And so you can see that for sample A, the TET-L gene has this very consistent SNP pattern indicated by the red vertical lines, and sample B does not. And so this is, again, a way to reconstruct the nucleotide level variation within the resistome across hundreds and thousands of resistance genes that might be in a given sample or data set, and to do that efficiently. So we've talked about scalability and accuracy, and the last thing I want to talk about is context. And so context is very important for the resistome. Because resistance genes are not all equal, right? So some resistance genes are going to be more risky to human health if they are, for instance, located within a pathogenic bacteria versus a commensal bacteria. They also will be potentially more risky to human health if they are on a mobile element like a plasmid that can be, as I said, shuttled back and forth between bacteria. And so understanding what we call the genomic context of these resistance genes is really critical for accurately assessing a risk of a given microbial community or given sample. However, it's very difficult to accurately co-localize or contextualize resistance genes within metagenomic data. And the main reason, is because we do fragment the DNA, sometimes multiple times, both in the lab and then bioinformatically. And we struggle to accurately reconstruct long fragments of DNA. So I showed you that we can do it with genes using the read color De Bruijn graph, but getting longer and longer, it gets less and less accurate. And so we showed recently basically that these existing methods for reconstructing AMR genes and their surrounding genomic material just don't perform very well and give very inconsistent results. Again, this matters because the level of risk is different, right? So if the gene is in a pathogen versus a commensal, if it's in a mobile genetic element, that has a different level of risk, and it's something that we would flag potentially in our AMR surveillance scheme as something to be concerned about. So our solution to this limitation was to take that enrichment approach that I talked about and apply it to long read sequencing platforms. And the reason we wanted to try this is because, of course, when you perform long read sequencing, you're maintaining the integrity of longer fragments of DNA all the way through the process. And so you no longer have to maybe reconstruct the original DNA strand, but instead you can just take it directly from each read. And so we wanted to see whether or not perform that enrichment would indeed work with long read sequencing, and whether that would give us contextual or co-localization information for the resistome. This is a very detailed overview of the workflow, but basically it's at a high level, it's fairly simple. So you extract DNA, you prepare it for long read sequencing, you do the bait enrichment, you amplify the captured DNA, you perform CCS sequencing. So in this study, we use the PacBio platform. We deduplicate the data, and then down at the bottom, we perform what we term co-localization. So trying to understand what's surrounding the resistance genes within each long read. So when we did this, we used a commercially available mock community with known genomes in actually known bacterial cells in known relative abundances within that mock community. And fortunately, another group, the Lohmann Lab, had published ultra deep non-enriched long read sequencing on the same mock community. And so that's represented over on the right-hand side. And so we were able to do, thanks to the open source nature of that work, we were able to compare the on-target sequencing using the TELS approach versus sort of deep and ultra deep sequencing of the same mock community without enrichment. And so despite much lower throughput, so when we sequenced these samples, we had much lower throughput than Nichols et al. Had in their data, but we saw also a significant increase in the percent on target sequencing, so a 15,000-fold increase compared to the deep sequencing. So this was promising. And then when we looked at the specificity of that bait enrichment, we saw that we not only had good sensitivity, but also good specificity. And the way that we did that is we took each genome within this mock community and we looked at the coverage profile of the target enriched data versus the deeply sequenced non-enriched data. And so that's what this is. So this is one of the mock genomes within that community. It's the E. coli genome. And this figure represents the full length of the genome across the bottom. And the areas of red are where we actually had probes designed to target the E. coli genome. Remember, we're only targeting resistance genes. So this E. coli genome has a lot of resistance sequences in it. You can see on the bottom with all those red spikes. And then the green spikes are where we had coverage from our TELS approach for one of our 8 KB library. And then the gray sort of throughout the bottom there is the deep sequencing from the GRID-ION from the previous Lohmann study. And what you can see in these call out regions that I'm calling out at the top is that where we have probes, so where we designed probes to target, the TELS sequencing coverage surpasses that of the non-enriched sequencing. And then in other areas of the genome, the non-enriched sequencing coverage surpasses the TELS coverage, which is what we want because those areas we weren't interested in. They did not have resistant sequences. So this shows us that our method is both sensitive and fairly specific, and indeed allows us to co-localize. So these are just a few examples of co-localization of resistance genes with important mobile genetic elements like plasmids and integrative and conjugative elements indicated in yellow, and then other cargo genes on those same reads. And so this information, like understanding that a CMY resistance gene is being carried on a given plasmid, is very critical from a public health surveillance standpoint. And so this is a promising result for contextualization. So hopefully you can see here that, you know, these three limitations, we are able to overcome them. Of course, there's always more work to do and more improvements to make. But we think that we've, you know, by and large, overcome some of these limitations so that we can generate a comprehensive, highly sensitive, highly accurate resistome profile. We can perform the analysis in a scalable, feasible manner bioinformatically, and then we can also contextualize these resistance genes to better assess risk. And so the overall result, I think, from these methodological improvements is that we're able to do comprehensive and efficient profiling of all AMR gene variants in a microbiome. And from a surveillance perspective, there's lots of use cases for this, right? But from a surveillance perspective, that allows us to perform comprehensive risk estimation for the samples that are being collected as part of the surveillance. And it also allows us to do highly resolved AMR tracking, right? So now we have accurate variant level information across hundreds of genes per sample. And that is a very powerful potential fingerprint to allow us to understand how these microbial communities are transmitting across all of these complex pathways. And so that's the hope, is that, you know, as sequencing becomes cheaper, and as we continue to improve these methods, that we can start integrating this microbiome-wide or metagenomic resistome approach into ongoing surveillance systems. So with the last couple of minutes here, I'm right at 44 minutes. So I think I'll do a couple of brief reflections. You know, the first thing that I reflect on when I do this type of work is that it's really cool and it's really fun to be able to move between like what we consider basic and applied science. And we often think of sort of starting with basic and moving to applied, but I think what is really fun is that actually becomes more bidirectional and iterative as you get deep into these topics, right? And we've actually had instances where we have applied questions more on the applied side, and we get talking to our collaborators about those, and it's like, oh, well, that actually has a really like cool basic science problem behind it, right? And so having that back and forth is really fun, and I think it makes these projects really strong. I also, you know, hope that you saw sort of this interplay between, you know, limitations and advancements in data generation and also data analysis, right? And those things, if they can be brought together by the right people at the right time and with the right amount of luck, you know, I think that again you can really get some strong advancements across the entire workflow. And to me, that's critical. Like you can't really always, you can generate some really cool and great new data, but if you don't have the tools to analyze it appropriately, you're not fully, you know, taking advantage of that. And so I think it's really fun to be able to do this. And of course, both of these things really require a team science approach. And so I'm just really lucky that I met some people who were interested in sort of the same areas, and they were willing to say, like, let's see what we can do. And they all have expertise in areas where I do not have expertise, and vice versa. And we can have these really great conversations where we each understand the limitations across the whole, you know, every field, and we can understand where we can make contributions and where maybe we can't make as many, but we can support, right? And so that's really fun. And I just really encourage everyone to think about that team science approach, especially when it comes to these really challenging things like antimicrobial resistance that do pose this global public health threat. So with that, I really thank you for your time. Of course, we also can't do this without funding. So we have funding from USDA, NIH, and then I'm a veterinarian and I work with livestock animals. And so we also have support from organizations that are interested in making sure that livestock production remains safe for human and animal health in the environment. And then, of course, all of my collaborators across multiple labs and multiple universities. I would not be talking to you today without them. So thanks so much. I'm happy to take questions.
Wonderful. Thank you, Noelle.
Yeah.
A lot of really, really fascinating stuff in there. And every time I wrote a question down, you then showed that you'd solved that question in your next few slides. So we have a few questions coming from the audience. The first is from Robert Blumenthal, which is, have your TELS data indicated whether particular bacterial species play a key role as vectors of AMRs, that they're particularly likely to carry AMRs between other bacterial species?
Yes, so that would take, so the TELS approach, we haven't published those results quite yet. And so it's really right now more of a methodology paper, I would say. We have since generated more data where we are trying to answer some of those biological questions, right? Like which bacteria are playing a key role in transferring certain resistance genes back and forth. But we haven't generated results from those yet. But the hope is that with this approach, those are the types of questions we can answer, right? Which bacteria are harboring which genes and which ones are moving them back and forth, potentially between pathogens and commensals in a given situation. That's where content matter, experts become really important, right? Like people who work on human diseases where resistance is a problem, are those resistance genes coming from the human gut microbiome? And how frequently and which commensal bacteria are the reservoirs if they're there, right? So those questions are all really why we're developing this approach. Yeah, and we hope to answer them.
Great.
Yeah.
Another question from Etienne Fafard-Couture is, what are the main approaches to identify novel or emergent resistance genes?
Yeah, so one reason that, the original way a lot of people do resistome analysis is using alignment. It's faster, it's less memory intensive than building that De Bruijn graph, which is an assembly-based approach. But one of the limitations of alignment is that you need to have a pretty close match to like known resistance genes. So you're really not going to find genes that are highly divergent from known resistance genes. So one reason we use the De Bruijn graph is because we're more tolerant in the De Bruijn graph of that sort of potential mismatching between known genes and novel genes. We've also improved upon that by using hidden Markov model profiles of different resistance gene classes. And what that allows us to do is to say, this locus of this resistance gene is critical for it to function as a resistance gene. And so we cannot have variability at that locus, but across the rest of the gene, we can have a lot of variability. And so by using that variability profile in the hidden Markov model, we are able to maintain sort of accuracy to say this probably has resistance function, even though it's highly divergent from known references in other areas of the gene. And that was published in Nature Communications a couple of years ago, that HMM approach.
Cool. And I actually have a quick follow-up question on that. So if you're looking at these loci that are really important, how long are they typically? I'm wondering if you could have, like, say, size selected paired 2x300 MiSeq reads and get enough there,
Yes.
without a long read.
Yeah, sometimes they're literally a single SNP. So some of those beta-lactam, like the ESBLs and the carbapenems, which are very important resistance genes from a clinical standpoint, some of those you really require like a given SNP or maybe a few SNPs. They can be dispersed across the gene, so that might be a challenge. But sometimes it just takes a couple SNPs to make it more resistant or to turn it into a resistance gene. In other cases, it really is longer loci across the gene that give it the function. So it really depends on the mechanism of resistance. Yeah.
Okay. Another question from an anonymous attendee is just wondering if your problem of detecting new variants in AMR genes is somewhat similar to finding new variants in tumor samples containing multiclonal cells. Can some methodological approaches used in cancer genetics be useful here as well?
Yeah, so I would say yes. So when we do resistome analysis, I think that there are areas that it's very similar and areas where it's different. So when we do a resistome analysis, we do have groups of resistance gene. We call them genes. They're honestly like alleles, right, of the same gene. And they're highly homologous across like 98% of the gene. And then you do have these areas of, you know, SNPs. And so in those instances, it's very similar to, like, I'm not a cancer person, but like what I understand to be these sort of mixed cancer tumor cells, right? And in other instances, though, it would sort of be like, then you have a totally different cancer in the same person, also with a bunch of variants, right? So because we're looking at the metagenome, we have like clusters of genes that are highly similar with very important variants, and then other clusters of highly homologous genes that are different than that cluster. So yeah, there are parallels, but it's even more complex, I think, because we're dealing with like thousands of different bacterial genomes and thousands of highly different genes.
Okay, and another question from an anonymous attendee: from a surveillance perspective, how does the cost of sequencing the resistome compare to culture-based techniques?
It's much more expensive, for sure. And so that's one of the limitations I did not list, because I have not made any contributions to lowering the cost. But what I would say is culture-based approaches are, they're more laborious, I would say, on the upfront, whereas metagenomics maybe is more laborious on the downside at this point in time in terms of the computation. The other thing I would say is, when you think about metagenomics, there's other things you can do with that data, right? And so you start to think about efficiencies just from the data set itself. Like, you can look at other things other than resistance as an example. And so I think that these techniques are complementary. Certainly whole genome sequencing also deserves a mention when we're talking about AMR surveillance. So most of the public health regulatory agencies are moving towards whole genome sequencing of these isolates, these pathogenic or indicator bacteria, and then looking for resistance genes within the genome. I think metagenomics plays a role in the way that I talked about in this talk, but it's not the only thing we need. And from a clinical perspective, that culture-based approach is really critical, because your doctor, if you have an infection, needs to know, like, what's the etiologic agent of that infection and how can I treat it most effectively. And that right now is definitely a culture-based thing. Yeah.
Although culture takes a while.
Yes. Yeah, so totally.
You get the sequencing done fast.
Yes.
That could
absolutely. I mean, like, culture independent diagnostic, technologies are tests are really also critical. And so again, I think and sometimes you use culture to confirm, what you found on a culture independent diagnostic test. But these things, for a while at least, I think will probably exist in tandem, which is good in my opinion. Yeah.
Great. One thing I was thinking about is, I'm still not sure how much you're missing when you're doing short read data, but it seemed like you got reasonable profiles as long as it was deep. And I'm wondering if you could use cross-sectional data, like go back to the HMP shotgun data, and use the co-abundance of bugs across people to gain an understanding of, who in healthy individuals, like which bugs are harboring which genes in a healthy population.
Yeah. So do you mean like using existing data or generating new data?
No, yeah, existing data. Just go back, pull like all the deep HMP shotgun data, use the short read data with alignment and your tools to profile the resistome, but then also profile the taxa, and based on how which genes are correlated with which bugs across people.
Yeah, so people have done things like that, sort of like a meta-analysis of existing data or multiple studies, right, to try to understand differences in the resistome profile. They've done that using, I think, HMP data, and then also like similar data from a cohort of people, I think in China. There's been some studies about that. That piece where, you're trying to correlate with abundances of bacteria. So the paper that I showed briefly with like those Venn diagrams that was just published in 2020, what we showed there is that if you use an alignment approach and then an assembly approach and then a different approach on the same data set, and you try to look at that sort of, it's not colocalization, but correlation, you get totally different answers depending on which approach you use. And in my mind, that's a problem, because the way that bacteria share these resistance genes, I don't think you can always say that like differences in abundance that correlate necessarily mean that they're linked genetically, if that makes sense.
Sure.
Yep.
Do we have a sense of what's a healthy resistome? And
No.
can you measure that,
Yeah.
either in humans or in wild animals?
Yeah. It's a great question. I think one of the things that was surprising very early on when people started looking at the resistome was just how much of it there is in like a given sample. There's just a lot of resistance genes out there. And what is the quote unquote baseline supposed to be? We don't know. People have, they've isolated like modern day resistance genes from like the stomach contents of, let's see, the Iceman, so what is anthropogenically driven, what is driven by antibiotic use, and what is sort of there already is a very difficult question to untangle. And so I don't think we have a good sense of that yet. Maybe we never will. I mean, part of the goal of surveillance is you got to start, right? You got to start like looking over time how trends are changing, because you can't always reconstruct what is the baseline supposed to be. Yeah, sometimes you have to just put a stake in the ground and say, this is the baseline, we're starting now, and we'll look forward in time, right? So
yeah.